funded by Gomery, so I'm a part of the USM Gomery Concord Consortium, and that's how I got to meet Sabrina. And after working with her in this project, I thought she would be a very good speaker for Dolphin Island, because she does really cool physical stuff, but she's been able to blend that in with a lot of interdisciplinary work that we're doing in that consortium. And so for a talk today, I thought all of you guys would be really interested in some of the interdisciplinary stuff that she's involved in, and I am really very interested in the physical stuff she does. So I thought she made for a very good person to bring to the C Lab. So I was excited to get her here. A little bit about her background is that she spent a lot of time at the University of Florida. She got her degrees in basically estuarine and coastal physical oceanography. I don't know exactly what the specific title is, but she does a lot of coastal and estuarine physical oceanography. And so today's talk is going to be on none of that, which I see some smiles there in the background. Today's talk is on actually some zooplankton work that she's been doing in our consortium. And when she first suggested looking into what she's doing, I remember thinking in the back of my head that this is a deep, dark hole that she may never come out of. And so in some recent meetings that we've had, some of the results she's getting are pretty wild, and I was really surprised and impressed by the work she's doing with the zooplankton stuff. So that was maybe a long-winded introduction, but I will turn it over to you.
Thank you. Thank you so much, Brian. Can y'all hear me? Yes? Cool. Well, that was a great introduction, but I have an introduction of my own. Let's start at the beginning. I was born in Puerto Rico, a little tiny island over there. That's me. In '86, and I've been slowly moving northward, but hopefully it ends around here, because I don't do so well with cold weather. As you can tell, I'm wearing a scarf in the warm winter day like today. So then after that, like Brian said, I moved up to Florida, went to the University of Florida to do a civil engineering degree for a bachelor's, and then went to grad school to do a PhD in coastal engineering, studying submarine springs in the Yucatan Peninsula of Mexico with Arnoldo Valle Levinson down there, which was a lot of fun. And then I decided to stay there. Why not? I've already been there for 10 years. Might as well do another year at the University of Florida with a postdoc studying the inner shelf circulation at Cape Canaveral, which is this little bump in Florida right there. And then I was really lucky because this whole Gomery thing gave us lots of money for Gulf research, so I am currently at the Naval Research Lab as a American Society for Engineering Education postdoc fellow. And my focus is to look at mixing and circulation on the shelf here in the Gulf of Mexico, and looking at the river discharges from the Mississippi and the Mobile Bay. So that's a little background, and I want this to be kind of like a feedback, kind of collaborative presentation. So if y'all have any questions or comments throughout my talk, feel free. I will reward you for good questions too, so keep that in mind. All right, so we're here today to learn about zooplankton diel vertical migrations in the Mississippi Bight. So apparently a lot of you are not physics people, right? So this will be an interesting combination, me being a physics person, not really knowing much about zooplankton, and then all of you experts out here. So treat me kindly. All right, so what are these diel vertical migrations? As you know, they're daily cycles of these organisms where they go down after sunrise for safety, supposedly at the bottom, and then during the nighttime they come up for food at the surface. But you can also have a reverse migration where they do the opposite. It's believed that these migrations are affected by changes in food or predators, the lunar cycle, or even tides, sunlight duration, like seasonal differences between winter and summer, stratification, and even like winds, currents, and turbulence. All of those could have an effect on these migration behaviors. And then for what my project is for, it's funded by the Gomri group, and I'm part of the Concord Consortium. So we're kind of trying to tie things in with that BP oil spill and try to understand how things relate to that. So these migrations could increase oil contact and advection of oil. So if the oil is at the surface and the zooplankton interact with it and they come down, then it moves oil from one layer to the other. All right, so now that we know kind of like the background, what kind of data did I have available to me? All right, so here is the Gulf of Mexico, and I worked in the Mississippi Bight. So if we zoom into this red box, Mississippi Bight, we're up here, very exciting. And then my data that I'll show you today is from this red box, and it includes all of these different colored dots. And I'll explain what each one means. So first, we had 5 bottom moorings, which is those blue dots, and they look like this. They had an ADCP in the middle, which is a acoustic Doppler current profiler. It just gives you profiles of current over time. And I'm going to show you vertical velocities and backscatter data from these instruments. They were deployed for around 6 months between November 2015 and April 2016. And there were 5 of them. 4 of them had the same configuration, and then 1 of them was measuring in bursts, so I have data of every hour. But I won't go into the details of the ADCP measurements. In addition to the ADCPs, we had plankton samples, net samples out there, which are represented by the red dots in this map. And they were performed at the beginning of the mooring deployment. Both day and night measurements were done at 3 discrete depths or parts of the water column, so near surface, mid-water, and near the bottom. With this instrumentation right here, and the samples were sorted and preserved by a great team of people from my Concord project. In addition to the nets, we observed zooplankton using the in situ ichthyoplankton imaging system, also known as ISIS. It's an unfortunate name, but it was done before this whole business. Anyways, they got to keep it. Can't let them win. And for what I'll show you today is tows that were done day and night, also at the beginning of the mooring deployment, using shadow graph lighting technique. So it just shines a light in, and the images that it takes is just the shadow of whatever is in between. And with that, it can detect all sorts of things, sizes of 400 micrometers to 13 centimeters. The towing speed of the instrument is 2.5 meters per second, which is relatively fast. But for this work, I'll just be focusing on organisms that are greater than 1 millimeter in diameter. And a colleague of mine went through the arduous process of sorting 50,000 images manually and classifying them by major species. So in addition to the ISIS, I'll show you data from a glider that was deployed during the 6-month of the moorings. It was only out there for about a month at the beginning of 2016, and I'll only show you data from about 2 days. The green line is the segment that I'll be showing you where the glider came within the mooring region. And I'll show you profiles of salinity and temperature for that to understand the passage of the Mississippi River plume over the moorings. So today I'm going to show you backscatter and vertical velocities, but I'm going to filter them. I'm going to take out all the periods that are longer than 36 hours to stay within that 24-hour window, because that's what we care about, the daily migrations of the zooplankton. And then I'm going to show you how they relate to sunset and sunrise, because that's when we have these drastic migrations at sunset and sunrise. So we'll see how it relates to that. And then we're going to look at what the net samples and the ISIS observe of zooplankton that are looking at the ones that are most likely to produce the scattering and the backscatter. And from the ISIS, we can also look at the behavior of these zooplankton, so we'll dig in a little into that. And we're going to use this really cool statistical analysis. It's very cool, all right? And we're going to use it to separate the variability of the daily cycle. And we're going to kind of dig deeper into those results. And then finally, we're going to see how the plume might have interacted with these vertical migrations. So first, we're going to look at the filtered backscatter. Here on the x-axis, we have days of the month. Each row is a different month, starting in November, going down to April, and the y-axis is depth in meters. And so clearly from this filtered backscatter, you can see that there's this like daily cycle, right? There's strong red segments and then there's strong blue segments, and that just means that there's higher backscatter intensity in red versus blue, which is a low backscatter intensity, and it changes every day. So, but don't take my word for it. We can zoom into this little segment, and I've kind of, the x-axis is still days. I've marked where sunrise and sunset are in these colors of orange and blue, and I've labeled what nighttime would be. And so clearly, nighttime we have increased backscatter intensity that sharply changes at sunset and sunrise, right? And we can even see these downward movement of backscatter right before sunrise. So clearly there's a relationship between what the backscatter is doing and the sunset and sunrise times, which tell us that it's a biological thing instead of tides or something else that might be more of a physical phenomenon. So to get a better idea of what these relationships between sunset and sunrise, I took every one of those months of data. I got an average of one day look like for the backscatter. And I did that for every month, which is what we have here. This is for the C1 mooring, which is down here on the bottom left, sorry. And then I've marked what the average sunset and sunrise times are for each month in those dashed lines. And so clearly there's this consistent pattern throughout the entire deployment where we have higher backscatter at night, and then we have this sharp downward movement of the backscatter right before sunrise, and then low backscatter during the day, and then high backscatter again after sunset. So this is a definitive pattern. So there's these diel migrations throughout the deployment. It's very exciting. And you don't, okay, so these are so you see these downward migrations here at C1, but you most clearly see them at the other moorings as well. And then, but sometimes after sunset, you also have these downward looking migrations as depicted by the black arrows. So there's some interesting stuff that probably represents a reverse migration. And then I did the same thing with the vertical velocities, put them in the same average of one day for each month with the sunset and sunrise depicted, and it shows what we expect, right? So downward movement, blue means downward, yellow and red means upward. So downward movement right before sunrise at night, and then during the day we have an upward movement, which is kind of strange, right? Because we didn't really see high backscatter during the day. So what are these upward velocities talking about? Well, it's something to look into a little later. And then at sunset, we have this very strong upward velocities, which goes with our idea of a nocturnal DVM. But then we also have these very strong upward velocities at sunrise, which would represent that reverse DVM. And the same pattern is observed throughout, so it's a very consistent pattern for this region. So then the question is, who's migrating? Who are these migrators that we're observing in the backscatter and the vertical velocities? Well, that's a good question. I'm glad you asked. But we can figure it out by looking at the net samples first, right? So they collected 4 different nets at 4 different locations. They ended up being 3 of them were during the day, which is these sections, and the grayed out section in the middle is nighttime, which we only had 1 net sample at nighttime. And we ended up having 3 main suspects in the net results: Keaton gnats, copepods, and ostracods. And these are the results. So for the Keaton gnats, we kind of have these circles represent the relative concentration for that sample. So in this first one, we have a little bit more Keaton gnats at the bottom than at the top, because these circles the circle at the bottom is bigger than at the top, and the little cross represents the average depth of these concentrations. So since it's somewhere near the middle, it shows that they're very spread out. They're not like concentrated in one depth. And then at night, we kind of see them move up a little bit, but the pattern, the DVM pattern, is not clear in these results. And then for the copepods, we have kind of a similar pattern where we have them pretty well distributed during the day, and then at night maybe they're a little bit more up in the water column, but it's kind of hard to say. And then for the ostracods, forgive my pronunciation if I'm like butchering all these words. It's not my language, but I'm trying. So for the ostracods, we have more in the middle of the water column, and then towards the bottom at this last daytime measurement. So what we know about the nets is that these are the most likely migrators. The migration patterns are not really there, but it's probably because we need more data, right? So if I was a net person, I would tell them to get a lot more samples during this 24-hour period. But thankfully, we have more data from the ISIS. Oh, that's a cute little copepod. So the ISIS did daytime and nighttime samples over here, and they do a bunch of profiles. So we can get a high resolution of the vertical distribution of these zooplankton. And so for the ISIS, we found out that the main zooplankton and likely migrators were hydromedusa, shrimp, and Keaton gnats, which are pictured up above over here. And in the daytime, we had the Keaton gnats being the most plentiful ones, kind of evenly distributed, but with less at the surface, while the shrimp were mostly within the top 3 meters of the water column. And then the hydromedusa had a similar pattern to the Keaton gnats, but with less concentration. But then at the nighttime, it changes. So we have the Keaton gnats going up towards the surface, and then decreasing with depth, the similar pattern with the hydromedusas, and then the shrimp also got away they got away from the surface in the daytime and were more spread out. So they have a reverse migration, while the hydromedusa and the Keaton gnats, I like to call them Cheetos, the Cheetos and the hydros, they seem to have a more of a nocturnal DVM pattern. So that's exciting, right? We kind of seem to have our likely suspects. But with the ISIS, we can also look at their behavior by looking at the orientation in the images. And we can, the relative angle of the orientation of these elongated zooplankton relative to ADCP could change the backscatter intensity, because so if the organism is horizontal, then the surface area as observed, the cross-sectional area as observed from below, from the ADCP, it's bigger than if it's vertical, right? It turns into a little dot. So we do care about what the orientation of these organisms is relative to the ADCP at the bottom. And we can do that by measuring the angle of their orientation. But if they're too round, they're not really going to change very much in their cross-sectional area. So we gotta look for the elongated zooplankton, and we fit an ellipse to their shape, and then from that we measure the angle from the horizontal, like this blue line over here, and that's the angle that we use to determine the orientation, right? So if there's 0 angle, then it's horizontal, 90 is vertical. And we can do that for the echidnats and the shrimp, because the hydros are not elongated. So from our results of calculating these orientations, we found out that for the echidnats, during the day, they're a lot more vertically oriented than at night. In the x-axis, we have the orientation. So 0 is horizontal, 90 is vertical, and then we have depth on the y-axis. These more green colors represent a higher percentage of the observations. So during the day, the echidnats were more vertically oriented throughout the water column than at night. These are less strong green colors, right? So that's pretty exciting, and that could be related to them, just as the ADCP sees them differently if they're horizontal and vertical, maybe predators see them differently. And if they're attacking from above or below, they're harder to see if they're vertically oriented. But that's just one theory, and it's a little bit beyond the scope of this talk. And then for the shrimp, it was even more noticeable. You know that they were concentrated in the top 3 meters of the water column during the day, and their vast majority of them are vertically oriented during the day, and then at night, pretty spread out. It's kind of hard to know what their preferred orientation is. So this is also something to consider when you're looking at the backscatter and trying to understand the DVM patterns. Maybe if you measure between day and night, they're oriented differently. So you may think that there's more or less in one at the day or the night, and maybe they have the same amount of concentration of zooplankton out there, but they're oriented differently, so your backscatter is different. So it's very important finding, and it's something that should really be considered when doing like backscatter to concentration estimates. So going back to the backscatter anomalies, from here we see that there's this clear daily signal, right? But it also changes. It has a lot of variability. Like up here, there's not a lot of that daily signal. It's not very strong, and then down here it gets pretty strong. So there's these pretty significant variability in the signal. How do we quantify it? Well, to really get an idea of like how different these things can be, here I have the mean of the whole period, 6 months. One average day of the backscatter at C1, which is this one. I'm pretty much focusing on that mooring. So it shows this signal that we're talking about, higher backscatter at night, lower backscatter during the day. But when you look at a specific day, it can be very different, right? You have all these different finer structures, more more variability in the signal. It could look like this, which is just a line coming down, or the whole water column is lit up. So there's a lot of differences in the signal, and a good way of quantifying those differences is by performing empirical orthogonal functions. And it has a really cool name, but we're just going to call it EOFs, right? And you're thinking, these are a lot of big words. I don't like them. I don't like them either. All right? But what are they? So let's say we're at a concert, right? It's pretty cool. And then you have in the crowd, right? You have people waving their arms to the music, right? You have the really fun and very dangerous crowd surfer guy, crowd surfing. It probably does it every once in a while. And then you have people shaking their butts depending on the beat of the music, right? So you have all of these different behaviors in the crowd throughout the whole concert. And it might, maybe like good songs are butt shaking songs, and you're like, yeah. But then other songs are more crowd surfing songs. Well, with empirical orthogonal functions, we could take all of those different movements, and we can try to separate them. And now we can say, all right, let's only look at hand waving. So we go look at only hand waving, and we can tell where it happened in the crowd. Maybe certain sections are more hand-wavy people than other sections. And then we can also tell during what songs they were moved to shake their hands up in the air, right? And we can do that for all three of the different modes of dancing going on in the crowd. So that's kind of what this EOF does. It takes all of this complicated motion in the ocean or at the concert, and it separates them into these more manageable modes of variability, and then you can focus and see what's going on. Does that make sense? Hopefully. Okay. Well, we're gonna see how the EOF worked for my data. So up here, these are the EOF results. I'm going to go step by step through them. So up here we have the mean, the entire mean of the backscatter, which we've already seen, same thing over and over again. And then below is the mean of the vertical velocities, which is that similar pattern we saw in the monthly means before, with downward velocities at night, upward velocities during the day. And then we apply the EOF signal, and it tells us about the variability. So we take out the mean, and we're looking at how things change over that period of time. So we have mode 1. Mode 1 represents 53% of the variability of the entire signal, and it looks very similar to the mean. So it means that, and then down here is a time series. So its x-axis is time, and then y-axis is the amplitude, which is used to modulate this mode depending on how strong it is. So when you have a strong positive mode 1, it adds to the mean. So it strengthens this DVM signal, this nocturnal DVM signal. And then when it's negative, it takes away, right? So it becomes a weaker DVM signal than this. And so throughout this 6-month period, you have some positives in the blue line, which is that blue color representing mode 1, and then you have some negative periods, right? And then we have mode 2, which represents 30% of the variability. Now this one looks more like a reverse migration, right? You have higher backscatter intensity at the bottom, and then it seems to go up at sunrise, stay up at the surface during the day, and then come down at sunset and stay at the bottom. So that seems to be like a reverse migration in mode 2, which is 30% of the time. And then mode 3 is 17% of the time, and it's mostly this like downward movement of the backscatter right before sunrise in the morning. And then these are the time series for all three. So I threw all this stuff at you But what does it mean? How do we really get a hold of what it is? So if we focus on this one day here in the middle of December, it seems like all three modes have a relatively similar percentage or amplitude in the positive direction. So let's look at how each of these modes contributes to that day's backscatter pattern. So at the top we have the backscatter. They're the same for December 16, and that entire day with time on the x-axis is hours and the local time. And then we have the mean, which is the same one we've seen over and over again. We can probably draw that from memory by now. All right, and then mode 1. Mode 1 on this day was had an amplitude of 0.3. So if we multiply that mode 1 picture we saw in the previous one by 0.3, this is what we get. And then mode 2 had a near 50 amplitude, 0.5 amplitude, and this is what that looks like for this day. And then mode 3 had a 0.3, which yields this. So these are the three different modes for this one day. And then if we add them all up together, we should be able to recreate what the backscatter looked like on that day. And that's what we do on this other column. So we start with a mean, we add mode 1, which as we learn, mode 1 amplifies that DVM signal if it's a positive amplitude, which it is here. And you can see there's a definite amplification of that signal between the mean and when we add mode 1. And then we add mode 2, which is that reverse migration, and it seems to add this higher backscatter at the surface during the day. And then we finish with mode 3, which strengthens this downward migration. So we end up having something pretty similar in the big scheme of things than the backscatter that we started with in the observations. So that's how this EOF stuff works. It separates all these different modes of variability, and then we can focus on each one and see how they vary. So that's what we're going to do next. All right, so let's focus on mode 1. Mode 1 here is the backscatter. I didn't really go into the vertical velocities, but they show an upward velocity at around sunrise and then downward velocities at sunset, which represents that reverse migration, but we don't really see it in the backscatter. And that could be because these velocities are observed by things that are not high scatterers, right? These are the loud ones that we see here in the backscatter, but probably these are not so loud, maybe like the hydromedusas, which are more jelly-like, so they would be harder to see, but still the velocities would be measured by the ADCP. So if we take mode 1, first things first, in the temporal realm down here, there's this clear upward trend in the blue line, if we focus on the blue line down here. So they start as kind of negative values at the beginning of our 6-month deployment, and they kind of make their way up, which makes sense, right? We have an increasing in intensity of this DVM signal from winter to spring, as you would expect. But also we have these other higher frequency oscillations in that time series. So what's happening there? Well, I think it has to do with the currents. So let's see what the currents are doing during this 6-month period. Here is a map of the moorings, the 5 moorings, and the black lines or ellipses are the ellipses of variability of the currents. So during the 6-month period, the currents pretty much seem to go back and forth like this in these ellipses in each one of the sites of the moorings. And the orientation of the ellipses tell us how the currents are oriented during that time. And it seems like most of the currents are parallel to the isobats, which are these gray lines which tell us the bathymetry depths. And it seems like the currents are just following the bathymetry in this area, with the red arrows representing the mean currents for this deployment, which are pretty teeny tiny, right? Relative to those ellipses, and that just means that there's not one way that the currents are going during this period. They go back and forth, and it seems to be going back and forth in a pretty consistent manner. So now that we kind of understand what the currents are doing, sound pretty good, right? We're going to look at C1, which is our favorite mooring, and we can kind of just look at this, the magnitude of the currents in this direction, which I'm going to call them the principal component currents for C1. And I extracted that time series, and then I compared it to the EOF mode 1 time series using a wavelet coherence. And that's probably my most favorite analysis method. Is pretty fun. If you want to talk about it afterwards with me, we can really dig deep into this method. But essentially a wavelet is like a spectrum, right? A spectrum gives you the peak energy at whatever frequencies. It tells you what the energy is at different frequencies of your signal. And that's what a wavelet does, right? But a wavelet is cooler because it tells you how that frequency changes over time. So sometimes maybe you have these strong frequencies of whatever period, and then other times you don't. So with a wavelet, you can actually see when you have stronger oscillations in your time series, which you can't see in a spectrum. And then you can take 2 time series, do the wavelets, right? You get these peaks everywhere, but you can now compare them. That's what a coherence does. It compares 2 time series, and it tells you when those 2 time series have similar energy at whatever periods. So in this case, the black line represents 95% confidence level, so anything higher than that is what we care about. And it says that when comparing mode 1 with the principal component at C1, we have high coherence within this black line, which is between periods of 16 and 32. So there's oscillations between 16 and 32 days in both the mode 1 and the currents. That's exciting, but what does that mean? All right, well, when you have a peak in the currents, you have a peak in the EOF mode 1. And then these arrows, they tell us the phase. And since they're pointing this way towards the left, they tell us that they're out of phase. So when you have negative currents, which would be currents going offshore, you have a peak. You have a positive peak in the EOF mode 1, which means you have a stronger intensity of the DVM nocturnal signal. And then when the currents are going towards the shore, so from the deep ocean to the shelf, then we have a lower or a trough in the mode 1 temporal amplitudes. So during this period between middle of December through April, we have this negative relationship where when the currents are coming from the shelf, we have stronger nocturnal DVM signal. It's pretty exciting. And that probably means that the waters that are coming from the shelf have a lot more zooplankton, or more zooplankton that likes to do DVM variability, or the DVM modulations. But I don't really talk too much about like quantity differences. I just really want to understand the patterns. So now that we understand what mode 1 does, we can move on to mode 2. Mode 2 is that reverse DVM signal. And if we look at the time series, it seems like it happens most strongly here in the middle of December. So what's going on in December that makes these mode 2 reverse DVM signal very happy? Because elsewhere it's negative or near zero, except here in December. All right, good question. So one of my super great colleagues, Inia Soto, she's collaborating with me on this. She's an expert on Karenia brevis and observing it from satellite data. And she helped me figure this stuff out. She has these methods of detecting harmful algal blooms from satellite data. So here, red means yes, there's a bloom, and blue means no. And for December, we can see that in early December, there's really not a lot of red in our Mississippi Bight region. Then in mid-December, it really blooms. It's a big party going on right there, especially over the moorings. These little white dots are the moorings. And then towards the end of December, it kind of fizzles out. And you can also look at the fluorescence line height, which kind of tells you the intensity of the blooms, and it tells the same story. There's not a lot going on before. Then in December 15, it really gets really intense, and then it dies down towards the end. So if we look at, now we know that there is a Karenia brevis bloom in this site around that time when we see that reverse migration. But what does the backscatter tell us, right? Well, here it is. These very thick black lines are these dates, December 8, December 15, and December 19 at midday for that day. I put them down here in this plot. And it shows that at the beginning, when the bloom is not really doing very much, the DVM signal doesn't seem very strong. It's actually extremely weak. You can barely see anything going on. And then towards December 15, it really picks up, it intensifies. We have strong, high backscatter intensity values in the bottom half of the water column at night, which is, these are in local time, so these tick marks for the days represent midnight. And then these dashed lines represent sunset and sunrise. And then during the day, we have an increase in backscatter at the surface, and then a huge void towards the bottom half of the water column, and then it repeats. And that is a reverse migration. But actually, K. brevis does a reverse migration. Previous work has shown that they stay at the surface within the top 2 meters or so during the day to grow their bloom, and then they come down to between 10, 20 meters in the water column at night. So which might explain what we're seeing here, actually. So bear with me. So at night, the high backscatter sections are below 20 meters. And that could be that these zooplankton are trying to avoid the K. brevis, at least the ones that are scattering. And then when the Karenia brevis goes up to the top surface, we have them go up towards within the top 20 meters, and then they come back down. So it could be that they're trying to avoid K. brevis, but somehow there might be more food or something that causes there to be an increase in the backscatter during this period. And this is the first time we're looking at the whole water column of backscatter during a K. brevis bloom, so it's also pretty exciting stuff for that work line of research. So now we understand more of that reverse migration. What about mode 3? 17% of the time, these intense downward migration at sunrise. What causes it, right? It's only 17% of the time, but there's this, what seems to be maybe like a monthly oscillation in the time series. At least that's where I started to figure this out. I looked at the yellow line, and I'm thinking, something is going on here. So I zoom in. This is a zoom in of the yellow line mode 3 time series, and I overlaid it with the lunar cycle, which is that blue line. And the high values represent full moon, low values represent a new moon, and there's this very direct and clear relationship between new moon and low mode 3 values. And then new moon and high mode 3 values, full moon, low mode 3. And what does that mean? Well, we go back here. When we have a new moon, we have high mode 3 values, which means an intensification of this downward movement of the backscatter. And that could be because of the light differences, right? If light is an important factor in the zooplankton moving up and down, because as we saw, they really are affected by sunset and sunrise, then when we have a new moon, we have a pitch black night. And then as the sun starts to rise, we have a sharper increase in sunlight. Maybe that's why there's a sharper downward movement of the zooplankton. Rather than in full moon, where it's a pretty well lit night, right? They might start migrating at different times and not all go down together to show this strong backscatter streak. So that's what mode 3 represents, is that lunar variability of the DVM signal. In addition to the EOF analysis, I also wanted to see what the Mississippi River plume does to these backscatter and the vertical extent of the DVM signal. So we had the glider traveling around here in this green line near mooring C1, which is our favorite mooring, right? And it showed these temperature and salinity profiles showing that at the surface, at the beginning of these 2 days of data, we have a thin Mississippi River plume, and then it kind of deepens as you get into the 3rd of February. And you can see that in both the temperature and the salinity profiles. But how does that interact, or how does that affect the backscatter and the DVM signal? Well, here we have the backscatter during those days, and what we see is 2 things. First, this is what first popped out at me. The vertical extent of these strong downward movements at sunrise seems to be here on the first day. It seems to be further up in the water column, just as the plume is further up in the water column. And then as the plume gets deeper, it seems to have kind of suppressed that vertical extent of at least these migrators that are coming down quickly at sunrise. So that's one thing. But you're going to tell me, what about this other stuff over here? The rest of the night, it matters too. All right, so if we look at these purple circles, there's not a lot of backscatter going on this day, at this night, throughout the water column when the plume is very thin and stable up there. And then as it gets deeper, it seems to have increased the backscatter throughout the water column, even within the plume. So whatever zooplankton we're looking at here, they have reacted favorably to this plume and have really lit up the water column. So there's definitely an interaction and like a multiple zooplankton species interacting here and having different DVM patterns, and that's something that, at least with this analysis, the EOF analysis that I showed you before, we're kind of better able to kind of separate some of these patterns from different migrators and dig in a little deeper to what's going on. But this is more qualitative, right? I'm only showing you 2 days. I had 6 months of data, but we only had CTD casts for a few days, and so next step would be to have a full time series of temperature and salinity with the backscatter to get a better idea of how these plumes affect the DVM signal. So what have we learned today? We learned that the ADCPs do capture the DVM signal of the zooplankton that happens throughout this 6-month period. The zooplankton, at least the cute gnats and the shrimp, oriented vertically during the day, which might have reduced the backscatter during the day, even though the concentrations might not have been any less. Currents transport the zooplankton and had an effect in increasing the backscatter intensity of the DVM signal when the currents were coming from the shelf. So that's exciting. The lunar cycle had an inverse relationship with the DVM signal, and harmful algal blooms amplified the reverse DVM migrations of shrimp and possibly other zooplankton. And then river discharges could work to suppress these DVM extent, the vertical extent of the DVMs, or even change their behavior. So these are some of the findings I have. My work, like I said, is funded by GOMRI and the Concord Consortium, and here are some of the references that I spoke about in my talk. Any questions? Thank you, thank you. Yes?
So what was the frequency you were using with the ADCP, and what size class of organisms would that correspond to?
Oh, yes. So 4 of the ADCPs had one measuring scheme, and those were 300 kilohertz. ADCPs that measure, I believe, like 1.25 millimeters diameter particles, and then the 600 kilohertz ADCP measures twice as big, so 2.5 millimeters. The C4 was the one that was the 600 kilohertz. Yes, good question. Here, have some candy. Yeah.
So
Another, yes.
So that's probably like mice and shrimp or something. Is that like, can you narrow down based on that size range what it is that you're...
Yeah. Yeah, well, the ISIS, definitely you can measure sizes, and the net samples, it's a little bit more coarse in the observations, but I was trying to be a little bit more qualitative in the analysis relative to the zooplankton because of the lack of zooplankton measurements throughout a 6-month period. But yeah, I think the size of the organisms is important, and it's probably within the ones that I have. Yeah.
So is there, kind of on that note, like I know there's been a lot of work on mice and other shrimp being picked up with this method. So with heating maps and